Hello, everyone. My name is Marie. I am your host for today's webinar. So, we are broadcasting live from Zurich. And for our guests for today, we have Ash, Hunter, and Sebastian, all also broadcasting live from Santa Cruz. So, we are very, very excited for today's session. I would say there's a lot of buzz going around on the topics that we have today, and looking forward to a very nice presentation by our guest speakers. So our agenda for today is as follows. So we are going to have an introduction by me about muscle biosystems and about high-density microelectrode arrays. And then we will go to our main topic, which is the talk from our guest speakers for today. After that, we will have our Q&A session. Then let me proceed with our introduction. So at Muscle Biosystems, we have next generation electrophysiology platforms that can be used for functional characterization of in vitro neuronal models. And our motto is every cell has a story to tell, and I can show you here why. First, about Muscle Biosystems. So we are a global technology leader in high content electrophysiology field. And already over 20 years, we have developed the technology that we have here at Muscle Biosystems, and we have top reputation in the relevant scientific domain. We have our headquarters here in Zurich, and we have our office in San Diego in the US. So we cater to hundreds of laboratories, whether that's academic laboratories or biotech, pharma, CROs around the world. And we provide expertise on the technology, hardware, software, as well as for applications. And I'm very happy that over these years, we have roughly around 300 publications by many of our users, and many of them also in top journals. So these are a few of those. And actually, for our speakers for today, they have recently published also their work in nice high impact journals, and we will let you know about that later. If in case you want to learn more about other publications, please scan through this QR code right here. Okay, so typically, there are very different in vitro biological models, whether these are neural or cardio, and they are in different forms. Like, for example, 2D cultures like iPSC-derived neurons or primary neurons, 3D like organoids or spheroids or assembloids, or acute samples like, for example, brain slices can be also from human and also retina. And across all of these biological samples, you could also apply them in different applications, for example, expanding them using microphysiological systems, or as what we will hear today, also for bio or neurocomputing. And we at Muscle Biosystems have traveled around the world, and we have listened to many scientists around talking about their different questions in the lab. For example, can I record anything from my samples? Are my results reproducible? Can I measure long term from my sample or from the same sample? Can I record hundreds of thousands of samples at the same time? Can I actually understand my data? And also, how can my data support my research question? So all of these are common questions that are recurring from what we have heard from many different scientists. And looking into this, we have seen also some current limitations in typical setups, whatever the setup is, regarding functional characterization. And these are the reasons why better measurement is actually needed for in vitro models. First is the noise. If you have high noise, for example, it could be difficult to detect any data. You need to wait longer until the signals are large enough before you're actually able to detect any activity. And at that point, only the large activity can be recorded and the subtle phenotypes cannot be detected at all. So it is important to consider the noise. Second, perhaps you do not have enough data points, whatever the method is. So it could result into low reproducibility of results. It could be also difficult for you to position your sample because you are limited in your recording area. And you could be missing out on a lot of cells because you just cannot record them. You don't have the capability to do so with your technique. And the third one is depending on the technique that you're using. It might be that, for example, especially for organoids, which is our topic for today, sometimes you would only be able to record acutely or once. You may not be able to measure it across multiple days, which could be relevant for some of the studies that some people want to do. And additionally, if you are not in the right environment, for example, inside of the incubator or a good environment for your organoid, it may be, the condition of the sample is unknown and it's not healthy, it will give you random results. So these are very, very important points. And at Muscle Biosystems, we really take these seriously. So what the field needs and what we are also inspired in all of our development at Muscle Biosystems are as follows. So we want to provide reliable functional data that supports translational confidence. That is low noise, high quality data, single cell resolution, and versatility for both being able to do acute and longitudinal recording for any sample type. And here we are showing organoid as an example also for today's talk. And not just that, this is mostly, let's say, for discovery. But if you want to even, scale up your experiments, you would need capability to, for example, connect to some sort of automation or even a way for you to handle large data sets and also analyze these data sets. And we're happy that not only us at Maswell, but also our users are also developing towards this direction. So we deliver all of these, and I'm going to show you now in the next slides. So first, insights from microelectrode arrays, and this is common from any microelectrode array. So here I'm just showing you ours, our Max1 high-density microelectrode array. And here at the sensor area, you can see the neurons as follows. And what you can typically record are spiking activity, like so, these traces, and then you can detect the spikes. You can then have an activity map of your sample where the active electrodes are or where the sample is located. And the resolution just depends on the technology that you're using. Here we have high density, so you can nicely appreciate the location of the cells as they are. And of course, you can also get the network dynamics because you have parallel readouts across many electrodes at once. So now, very specific and special for Maxwell Biosystems, we can extract single cell and subcellular metrics, which allows us to have high quality, high reliability, and towards high throughput for HD-MEAs. And what this means is that we can actually extract single cell activity. Of course, that's after some processing of the data. And here you can even relate, for example, the firing of specific neurons according to their types. And you can even dig deeper, such as the axonal information of your cells using our axon tracking assay, wherein you can really follow an action potential as it initiates and propagates along the axons, and can then extract not only the velocity of such propagation, but also some morphology of these axons, which you can then also again relate to the type of neuron, as here, for example, the cheer is something, a cheer and BMP is something like this, and so on. So how is this for multiple types of samples? So this can be done, of course, for all 2D samples. These are different types from primary neurons to different IPC-derived neurons, and also for organoids, but you'll hear more about this today. And all of these are possible by this technology. So when you zoom in into our sensor area, you can see this 2 by 4 millimeter square area, wherein there are in total 26,400 electrodes that can be used for recording and also for stimulation, and they are all low noise readouts. If you zoom in further, you can see the high density of electrodes, and the distance between the two electrodes center to center is 17.5 micrometers. So you can really capture the signals from single cells or even those that are propagating. And the important thing is that it's uncompromised as you go to multiple wells. So it's the very same sensor when you go to the 6 well and even to the 24 well. So you don't lose any information, whichever well format you're using. In terms of systems, you can use the single well, which is the Max1 system as shown here. It's really flexible, it's very small, you can combine it with many different other devices. You can even carry it on your backpack, or it's really like a size of your phone, and you can put it inside the incubator and do continuous recordings inside the incubator, which could be valuable, especially for the work that is going to be shown here today. On the other hand, if you want to go high throughput, we have the Max2 system, which is compatible for 6 and 24 well plates and even for our upcoming 96 well plate. So you just need a single system for these multi-well formats from 6 to 96. And yeah, so of course, very central for our offering at Muscle Biosystems is the software. And we have an all-in-one solution that allows you to visualize the data, record the data, analyze the data, and importantly for scaling up, exporting metrics and reports. And the different assays that you could do, for example, related to neural recordings are activity scan, network recording, axon tracking, and also stimulation. If you would want to know more information, just write in the chat and then we can talk with you specifically about this. We have the following for large-scale data handling. So we have the annotation capability so that you can then recall the information for every well that you have recorded from, if you added some compound or which cell type those are, and then afterwards do a batch analysis of all of your recordings and export all of those either in for example, Excel or CSV, or also in with the graphs here, which you can export in PNG, PDF, or PowerPoint. And then something also important for our talk today is the API, which you can use. For example, you can just program yourself the capability of the MEA accessing the stimulation and the recording, wherein you can use open loop approach, where you just design your stimulation and then read out the results, and then according to your algorithm, given other stimulation patterns, or in a closed loop pattern, wherein you close the loop by just recording, stimulating, recording, stimulating. And these are two papers, just example papers using open loop. So this is the reservoir computing on brain organoids by Fengua's lab in Indiana University, and a closed loop approach by cortical labs, where they use the device for playing or training the cells to play the game Pong. All right, now I'm going to introduce you some new things also from Muscle Biosystems, some accessories to support your research. So first, because we really support ease of use and also making sure that your results are reproducible, if you're doing cell cultures, it could be valuable that whoever is doing the culturing in the lab, it's always the same result. So we have this solution called dot plating accessory, wherein you can just place this frame. There is the spot where you can just lean in your pipette. You can add in your, for example, coating or your cell suspension. Even if you're transferring, from the hood to the incubator, it's not shaking out. It stays there, and it really makes sure that your cells are really centered to where the sensor is. And it's really designed for precise sample coverage of the chip. Very, very useful and also very nice to have in the lab. And this is not only available for Max 1, but also for multi-well formats as well. The second thing is for organoids. Of course, the sensor is sensitive enough to record from the organoids. You can culture the organoids on our chip, but if you would like to, for example, record it acute, we have this organoid holder wherein you can place the organoid on the chip, and then just add this membrane, which has, let's say, these flexible arms to ensure that your organoid is nicely attached onto the surface of the MEA, and it doesn't move around even if you're moving the MEA around. And it has very reliable measurements, and it's also very easy to handle. And also the organoid is very well healthy and protected. It doesn't get hurt, let's say, by the process, and you can take out the organoid again and record it again the next day or so on. So these are the new accessories that we have, and if you're interested, please send us some messages through chat and we'll be happy to reach out to you. Additionally, also, we do have compatibility for automation. So our Max 2 has the API for hardware and software control, and together with our large-scale data handling and report generation, you can integrate our Max 2 system in any automation solutions that you have in the lab. So once again, if you're interested, just let us know, and happy to chat about this. Okay, that's the intro. So at Muscle Biosystems, we have a focus on neurocomputing this year. We will be sending you tutorials on different guides on how to use our API, some blog posts on emerging trends. We also have webinar like today, user perspectives, and others. So if you would like to follow, all these materials coming on the neurocomputing related topics, just scan this or go to our website, go to resources, and then to neurocomputing, and you can find the latest information. All right, thank you so much for being with me for this intro. Now I'd like to introduce our speakers for today. There are three of them, And I'll introduce first for the first paper and then the second paper. So first is Hunter Schweiger. Hi, Hunter. He's a PhD student in the molecular cell and developmental biology at the University of California, Santa Cruz, and co-advised by Dr. Mohamed Wastayo Raji and Dr. David Hausler. So his research focuses on understanding circuit development in the brain through the lens of evolution and neurodevelopmental disorders. Another speaker is Sebastian Hernandez. He's also a PhD student in computer and electrical engineering at the University of California, Santa Cruz, and he's co-supervised by Dr. Misha Todorescu and Dr. Mohamed Wastayo Raji. And he's interested in understanding the interface between biology and computer to uncover algorithms underlying biology. So both of them are co-authors in the paper recently published in Stem Cell Reports, wherein they used mouse forebrain organoids to show how distinct excitatory and inhibitory compositions give rise to divergent network topologies. So you can hear more about that later in their presentation. And our third speaker is Dr. Ash Robbins. He recently graduated and it is now a postdoc in electrical and computer engineering at also UCSC, advised by Dr. Mircea Chodorescu. So his research focuses on developing closed-loop electrophysiology frameworks for brain organoids, combining artificial reinforcement learning with biological neural networks. So his recent work, another paper, was recently published in Cell Reports, demonstrating that goal-directed learning in cortical organoids is possible using the mouse brain organoids that is presented by Hunter and Sebastian. And outside of the lab, he's the CTO and co-founder of Emergo Labs, a VR platform for physical rehabilitation. So yeah, looking forward for the presentations, and the floor is yours, Hunter.
Thank you for that wonderful introduction. My name is Hunter Schweiger, and I'm excited to talk to you about our research with my colleagues Sebastian Hernandez and Ash Robbins. Our talk from self-organization to learning, mouse forebrain organoids as models of network assembly and goal-directed adaptation. And so the first part of the talk will be by myself and Sebastian Hernandez on our work showing these differences in network assembly by perturbing the excitatory to inhibitory ratio. And then Ash, in the second half of the talk, will be talking about the goal-directed learning in vitro. So I'd like to start with the question of how well do organoids recapitulate brain development? And so there's a large corpus of literature surrounding human brain organoids, but although we can match them and show transcriptomically that they're matching the in vivo tissue, it's difficult to measure the electrophysiology to ground the same observations that we would see in organoids. And so we can try and match to some in vivo trends where there's, again, a large corpus of literature from people like Peyman Golshani or Mattiacini in their respective labs showing that the in vivo developing mouse, that it has this progressive synchronization of the population activity followed by a decorrelation. And so then you can ask, well, if we have this large body of information surrounding the in vivo, well, then why would we need mouse organoids? And I would say that it is difficult to disentangle what is actually driving these developmental trends of the in vivo mouse because there are so many developmental processes happening at the same time. And so our work focuses on trying to disentangle these factors and figure out what is the primary driving force, and whether it's the interneurons that are migrating and integrating into the circuit, or it is the innervation from the other brain regions into the cortex driving this desynchronization. And so we use these forebrain organoids that use a classical TGF-beta and Wnt inhibition. And then very key for our protocol is that we are using this brain phys-based media that allows us to create these electrophysiological conditions that can better recapitulate the network assembly. And then a key is for the ventral forebrain organoids that we're trying to enrich in these interneuron population is this sonic hedgehog agonist, which in development is allowing for the ventralization of the tissue, where these interneuron progenitors are born and then will later migrate into the cortex. And so by adding in these factors, we can enrich for either what we show at day 10 is these interneuron progenitors are much higher ratio by this NKX 2.1 marker, or in the dorsal forebrain, the excitatory neuron progenitors by this PAC6. And what was cool is that at day 40, we saw that the ventral forebrain was much higher enriched for the parvalbumin and somatostatin interneurons. And particularly, parvalbumin is very difficult to model in human, as takes many, many months to get this interneuron subtype. And we got it in very abundant numbers, which was cool to see. We also perform single-cell RNA sequencing for characterizing the tissue, and by mapping our query cells from our organoids into this primary UMAP space, we can show that we are recapitulating all the same cell types that are in the developing mouse brain. And so by using the Maxwell, we characterize over 30,000 putative neurons about over a month of the development once the networks are formed. And so with that, I'm going to hand it off to Sebastian to talk about how we are characterizing the networks.
Thank you, Hunter. So, one of the metrics that we were really interested on keep understanding and analyzing in our data is the spike time tiling coefficient, which measures the probability of one neuron spiking when the other one is spiking as well in a window of time. Next, please. So part of the things that we do is, well, we calculate all the metrics and the matrix of the different interactions. Next, please. And some of the things that are really important to mention is that by mere probability or randomness, two neurons can be highly correlated even though they are not connecting directly. So in order to just improve the specific metric to avoid some kind of fighting rate bias, we performed this shuffling, which we had different like 1,000 shuffles on the different neurons on different time scales to preserve the population rate, but disrupting the different interactions that they may have. Next, please. Once we have this shuffling, we can generate the surrogate networks, and that surrogate networks, which are related to the parent network, which is the one that we are trying to measure itself, we can filter, and we use the 90th percentile threshold to really kind of filter out those that may be noise and just keeping the connections that may be the most significant one in those networks. Next one, please. So this is an example. We have the spike time tiling coefficient. And when we compare the dorsal versus the ventral, what we observe is something that kind of ties what Hunter was mentioning at the very beginning, that throughout the development, the mouse brain tends to decorrelate through time. And what we obtain is that dorsal networks, which are highly enriched in excitatory neurons, they keep increasing in synchrony, while the ventral, they were decorrelating. So that calls the attention that the inhibitory neurons may be the ones that are producing this decorrelation. Next one, please. So in order to keep characterizing some things that we were also interested was the network topology. So when we have regular networks, usually each node is connected to the other one. So there are long range connections, and usually the specific capability of the network is not that high because we don't have long range connections. In random networks, what we have is more like uh, higher connectivity differently between nodes, but they are not well organized. And the middle ground is the small world network, in which it has been shown that allows to have an efficient transportation of information through different networks. Next one, please. So when we measure this, we observe that both dorsal and ventral had different capabilities on the networks, meaning that they have a small world networkness, but also the ventral network seems to have a clustering coefficient that was higher than the dorsal networks. And that may talk about how different networks can have more local capabilities of information processing. Next one, please. Therefore, we apply this K-core algorithm that if we see it, it is like you have the popular kids and the non-popular kids, and you start basically taking out those nodes that are not so popular, and then you measure the network and you see what is left. And then you keep taking out nodes until you have a specific group of nodes that are highly connected among themselves. So those that were taken out are called the periphery, and the core are those kind of highly connected nodes that may be really important for driving the dynamics in that system. Next one, please. So when we perform this specific algorithm in dorsal versus ventral, something that was interesting is that ventral usually had a less ratio core periphery, meaning that the core was smaller and the periphery usually tends to be higher comparing to the dorsal networks. Next one, please. So when we plotted all these networks in the specific like space on the network connections, what we observe is the dorsal networks tend to be highly connected among themselves. Doesn't matter the specific stages, developmental stages that we chose. And the ventral usually have these kind of modules that are apart and a really small core that usually do not change throughout the development. Next one, please. So this talked us about that there are different connectivity patterns associated for each one of the organoids, dorsal enriching excitatory neurons and ventral enrich in inhibitory neurons. And that can also talk us about this modularity. So as we saw the different cores that we saw, the different modules, ventral network seems to be much more higher modularity. And these modules usually increases throughout the development, which is interesting because we may start thinking on a hypothesis that inhibitory neurons may be the ones driving this specific behavior in which they form modules and they may produce differences in the physiology. So next one, please. When we then see each of the modules and next one, generated all the firing rates, what we can see is that the population firing rates of each of the modules in the dorsal communities usually tend to fire all together, meaning that these modules seems to be highly correlated, versus in the ventral communities, these modules tends to be decorrelated, meaning that when for example, the orange one is firing, the green one uh, fires with some latent time. Therefore, that also produces some interesting characteristics that then Ash will explain a little bit more about. But just to finish, we measured this during, as Hunter said, different months and days of lives of different dorsal and ventral organoids. Meaning that this observation is significant, and that brings some questions about what may be the code of connectivity. And organoids are a great window to talk about this code. Now I'm handling the floor to Ash.
Awesome. So from everything we were just shown here, we can kind of extrapolate once we can kind of pull out some things around the connectivity. And just trying to dig into understanding these cultures in a different type of way, we asked this big question, which is just how can we actually use this electrical stimulation to characterize these functional networks and see if we can get them to do anything, modulate in certain ways, and just kind of learn a little bit more about them. Um, so my background is actually robotics and controls. So there's this one core problem that everybody learns in one of the early classes, which is an inverted pendulum problem. So here we call it cart pull, and it's this little cart that can move horizontally. And the goal is to try and just balance this pull on top of the cart. And the problem with this is that you have to control every single time. If you make any little bit of mistakes, it falls down. It's a really quick, rapid problem. And it requires continuous control to actually be solving this problem the whole time. So, of course, my whole goal was through most of my PhD was to basically get an organoid to be able to control this problem and see if it can modulate in the proper ways and get it to actually solve it. And pictured here. So how we kind of built this is we use this multi-phase approach to make this an automated experiment using the Maxwell. And what we have is this thing that will start with recording and trying to characterize the actual network, identify what neurons are. We'll go to stimulating those individually, and then finally we set everything into this big training period to actually put it into the task. Um, and with this, we want it to be pretty automated, pretty fast, because once we get the organoids, we want to kind of characterize them as they go. We don't want things to be changing too much. And yeah, we need a whole clean and analyzed process, and we use a lot of the Maxwell API with some tools on top of it in order to get this going. Um, so what does this actually look like? Okay, we take our organoid, we have it sitting on an array, we do one of our activity scans to pick out where the actual activity lies. We can get cool pictures of the activity, identify where specific neurons are, pick out like key hot points, and then from here we can kind of get into preliminary spike sorting to identify the actual individual neurons in the culture. So this is the whole like kind of characterization phase. Usually we do it at about 5 minute recording and we analyze the data right after. It takes less than 30 seconds or so. Um, then we get into what's actually stimulation. So this was before the whole axon tracking assay came out. This was a lot earlier, but using some stimulations, we can actually see that if we lay a few on top of each other, we could see the axons propagate from specific neurons, and we could see this like kind of probabilistically happening. And the higher that you stimulate, we can get a higher probability, and we can build these curves of how much amplitude you need to stimulate in order to actually get a positive reaction. So the giant thing over here is a stimulation artifact, and then after that, everything after in the following milliseconds is the axon propagation going on, which is awesome. So once we identify that stimulation, we can now say, hey, why don't we just hit each neuron a certain amount of times and kind of probabilistically identify what's going on. So we see some reactions that occur within the first 10 to 20 milliseconds sometimes here, and all the red is encoding the first spikes that happen after the stimulus, and this is 50 stimuluses on top of each other overlapped, and we can get some curves here, and then we can see a different type of activity where we see this like secondary longer term reactions or kind of more bursts happening and things along those lines. So there's a lot of different kind of characterizations of activities that happen once you stimulate. So what we did in our like kind of second part, so the first part was the recording characterizing. The second part is this causal connectivity or this stimulus evoked responses. So we can kind of split the responses into these two kind of shorter term interactions and then the longer term kind of reactions. And we can build these connectivity plots similar to the spike time tiling, but specifically from these evoked responses. And we can get some nice interactions of when you stimulate certain input neurons, how the output neurons respond. And we can get some longer term reactions as well. And we can also build a connectivity map in the same way. So in order to do this, we also wanted to basically identify which neurons we should put information into and which ones we should read information out from. So generally, we select ones that will evoke downstream activity as the input units, and the output ones would be the ones that are kind of more evoked. And then we pick separate neurons as actual training neurons as well. So what does this actual training process look like? So realistically, what we do is we take an organoid sitting there, spontaneous activity, has its dynamics going on. We read out from this activity. And we have some math of how we read out it, but it's essentially rate decoding. And we can take the activity from the organoid really quickly in the order of like milliseconds, we can read that activity, decode that into an actual force, and use that force to control the little cart that we were talking about earlier. Then from there, we can take the cart, see what happens, how it moved, encode the new angle that the pole's at as a frequency, stimulate that back into the organoid, also using rate encoding with a different formula, um, and then send that back into the organoid. And we do that all on the order of either 100 to 150 milliseconds. We could do it a lot faster as well. But yeah, so we do that rapidly and over again. And now we have a time step for how this works. And what that looks like is you'll get a cart pole kind of moving, interacting in the environment, but nothing super interesting is happening here yet. It's just attached to the organoid. It's not actually learning or optimizing in any way other than kind of natively. So for the paradigm we actually used, every time that the organoid or the carpal actually falls over, we consider that the end of an episode. And if it continues, it's balancing within the same episode. So when it actually falls over, we then have the choice of delivering one of these training pulses. And for the sake of the whole paper, we chose three different types of training pulses. One is just no training pulse at all. The other one is this randomly determined order of training pulse, of the stimulations in a training pulse. And the last one is this adaptive uh, approach. Um, and so what these actually look like, they're just biphasic pulses um, with and we have a small delay, 10 milliseconds between each neuron in the like kind of training pulse pattern. Um, and we deliver these training pulses only if the last 5 episodes, so when the carpal was going before it fell, um, we're not improving compared to the last 20 episodes. So we can see this on the red and green plots here. We see that they're only happening when there isn't improvement happening, essentially. And so the null, we have basically no stimulations going on at all. The random, we have this randomly organized selection of neurons for each training pattern. And then for the adaptive, we have these basically choices of pulses with specific orders to them. And we optimize over the adaptive with a reinforcement learning algorithm that tries to select the adaptive pulses in order to maximize the reward and all. So how does this actually work? When we run this, we basically see that the adaptive uh, has a lot of trials and tends to optimize over time, and we can get these like much longer balances that survive for a lot longer and are able to basically stay balancing. And for the null and the random cases, for random, we generally get a lot of more kind of chaotic ability of balancing or not balancing. And for the null cases, we tend to basically get no improvement at all, if not even a decrease in activity. Um, and if we look at any of these trials over time, we can see that this different way of like watching the dynamics. We can see that the in the adaptive case, early on in the like kind of bluish area, they will fall off and they'll have maybe one or two corrections possibly. But then towards the end of the training, we get to up to 10 to 20 corrections in the specific one. With the null case, we don't really see any adaptation there. And with the random case, we kind of get this chaotic, like a little bit early, a little bit late, but nothing consistent and continuous there. So from here, we basically took the adaptive case and decided to just basically let's train only with the adaptive case because we showed that this one, we demonstrated that it was able to work a lot better. And we created this threshold uh, of using some random networks in order to like see what the possible best average cases were that you could do with these random networks. And we took a few standard deviations out from that and called that a threshold that you couldn't really randomly learn past this point, no matter what. And we see that in the adaptive case, it kind of blows past this every single time when you continuously train, and that we get really good results using just this paradigm. But when we use this rest period in between, we do see that the activity resets down, but it's able to generally learn back up to the same kind of capabilities as earlier. And so if we look at one of these trials on a deeper case, we could see that early on, when we're looking at this space between the angle of the pole and then the output of the kind of the decoding neurons, so the like kind of ones controlling the motor movement, what we would expect here in a perfect solution is that we would actually see it oscillate around this space. We see early on it's not able to do that, but then later on we start getting these nice oscillations around this like phase diagram. Um, And it looks really nice, and it shows actually too. It's not balancing perfectly in the center. It's a little bit off center how it balanced, so it has a bias towards one of the sides, but it's able to kind of get a lot of oscillations around this, and that is a really beautiful result that came out. So really happy with that. Um, and you can also take this, we'll glance over this really quickly, but you can take this into the 3D case and look at it based off of the angle and the derivative of the pole as well. And what you would expect here is we get this really, you would hope for a nice split on the diagonal, where you see really high red activity on one and really low blue activity on the other. And if we watch this optimize over time in the adaptive case, we see it can kind of come out. So this is essentially showing the policy of how the organoid learns over time. So we see that the activity normally starts kind of neutral all across, and then as it goes on, we split into this separation of activity based off of the angle of the pole and all. Just kind of emphasizing that this network is optimizing to kind of solve this over time. And if we look at these different cases, we see based on the number of cycles on the left side, with the time balanced, it's not every single case for the adaptive that they work, but in the adaptive, we get ones that absolutely work and go way past this threshold of learning. And if we look at the right side, we can see that when we only train the adaptive, we get up to this like 45% time of being able to actually solve this problem, which is really cool. And to show that this required basically glutamatergic transmission, we used APV and NBQX, and we delivered this, and we see that we were able to kind of demolish the capabilities of the network, and then they would recover after a wash period here. And the bounds is a little bit difficult to see, but they're in the dashed lines for corresponding with each color, and these are 3 different organoids, one in each color. So they all recover back up to about the same activity as before. But yeah. And then finally, here's a little video of them going. So early on, we can see that it'll fall over and not really bounce too much. And then on the bottom, we can see late. And these are sped up a ton just so we can actually get some samples in that we can watch. But on the bottom, we can see it's able to balance in a way that it kind of like zooms across and is controlling the capabilities from falling over. And yeah, so any lapse of activity here, like you pick the wrong direction for even 2 or 3 time steps and you fall completely. So it's able to fully control. And on the left side of each of these graphs, we can see the angle of the actual pole. Um, so yeah. So the whole idea here is this is a basic essential controls problem, but this is kind of just the beginning of this whole new neurocomputer era. So we can do these automated recordings, these automated stimulation paradigms and analysis, and then even get the training automated, all that. So you can click a button and boom, go through all this stuff, select neurons, and kind of identify how things can actually train and modulate. And so this started with the carpool problem here. If anyone remembers from the being at the Maxwell conference, like I think it was 2 years ago or something like that, I presented on how a class was able to kind of work for and play around with coding this problem, but it's like a pretty complicated problem. But being able to get a class of students to write code for this and everything was really awesome, and seeing all them experience that. And then we've got a lot of future stuff with robotics and other things that can interface in this as well that I won't really touch on here, but it'll be coming out. Things around this whole paradigm. So from here, it's like we can interface organoids and neurocomputers with all sorts of different activities and cultures and read in and out information. And I think the whole field just needs to kind of get interacting with all these organoids and learning how they can actually learn themselves. So yeah, and that's it for me. So thank you all for listening.
Thank you so much, Ash, Hunter, and Sebastian. Congratulations to all of you. Really fantastic work, and yeah, nice results as well from both research. Now let's go straight ahead to our Q&As. Maybe a first few from me. What is exactly the size of these mouse organoids, and for how long do you culture them before you start recording or stimulation?
Yeah, the mouse organoids are about 1.5 to 2 millimeters,
Oh,
and typically the we culture them for about 30 days before we start recording. And then for the kind of mature network phenotypes, we don't really see the divergence until about day 45. It
mhm.
increases for a bit, but in that time we can start playing. They can start playing games as well. So start
Cool,
training
cool.
them
So
early.
mature quite fast then.
Yeah, that's one of the big advantages over the human, where we've started doing uh, more human cultures now and getting them to play games as well. And that takes many months of preparation. So, um, yeah, the mouse allows for a very fast iteration.
Thank you. So now I'm going straight ahead to the questions. So the first one is from Gillian McMahon. Hello. The question is about if you're measuring on one side of the organoid on a single plane, how are you capturing enough information to determine a network type? I think, Sebastian, you mentioned about the different types of networks, so perhaps you could answer this.
Yes, thank you. So, of course, it is kind of a limitation that we have from the system. However, what we are aiming is like to have like a representation, like a representative window into this kind of functional state that each of the units that we are measuring from are having, that in which the network type can be inferred from these population level features. So it is more like, we can say, even like functional network that we can read from the MEA rather than the whole network. However, in future works, we have been also like trying to understand how these networks are established through the 3D dimension. And in the paper, we also have like some 3D stainings showing
Mm-hmm.
that the organoids have like kind of an homogeneous network representation through the organoids. So yes, for sure, like from the functional side, we can only read those neurons. However, it seems that it may be applicable that the whole organoid also follow what we are reading functionally at that time point.
Thanks a lot, Sebastian. I'm now, because we have many questions, I'm going kind of quickly through them. It's kind of a related question from Janusz Vorosh. Hello, Janusz. Nice to have you here. He asked, what percent of the mouse organoid do you actually record, or maybe even how many neurons are you recording from at a time? If you know it, anyone would like to answer?
So I would say we get on average anywhere from 150 to 300 after high quality spike sorting and quality control metrics. Once an estimate of how many neurons are actually in the organoid total. If we dissociate them, we can get anywhere from like 1.5, 2 million cells. So you can record from a fraction of them, but that's a limitation of any electrodes and just the field in general. Yeah.
Thanks a lot.
Just adding a little
Yeah.
bit, like all these kind of network metrics are
Mm-hmm.
sensitive on the number of neurons that you may have. So we
Mm-hmm.
also filter out those networks that had less than 60 neurons. So
Mm-hmm.
if you have just like two neurons, of course, they're going to be like highly correlated, so on and
Yeah.
so forth. So part of the filtering and processing of the data was taking out those networks that didn't accomplished to have like a great number of neurons, so these networks analyses are more valid.
Yep, yep. But it's already impressive that you're getting actually these lots of neurons from the organoids. Thank you. I'm skipping now to Santa Maria Lab. So to go from number of time steps to total time balance, you used the organoid time step or the physical cart pulse simulation. So that's either 100 to 150 milliseconds or the 2 milliseconds. Maybe this is for you, Ash.
Yeah, we use the organoid time step for this one. Exactly. So the amount of time that the organoid was able to see the simulation. Yeah.
Okay, thank you. Um, Next, this is from Robam Zezewa. Hello, Ropa. She said, great talk. Quite impressive to see how we can distinguish correlation and decorrelation between neural region organoids. That's the dorsal versus ventral forebrain regions. So her question is, how adaptable is this algorithm to more complex models like assembloids? Are you looking into testing into this, or you have probably new models that are brewing in the lab?
We're currently interested on understanding how assembloids may be changing through the development while they are integrating.
Yeah.
Of course, there are challenges as we are talking about with the 3D because this integration happens in different stages. There is one that is transversal and the other one that is more like towards inside of the like cortical columns. This happens in vivo. In organoids, of course, like you will have like migration, but
Mm-hmm.
how can we measure that migration? So in this case, controls are the only way that we have to take out any confounding variables, but it is something that we're working actively in the lab.
Awesome. Thank you so much. Next, we have Frank Chen. Hi, Dr. Robin. So this is for you, Ash. Thank you for the presentation. I have a question about the adaptive training. From the plot, it seems that after resting, the performance go back to zero. Does it mean that the brain organoid cannot remember the training? Have you tested how long the training effects can last? Thank you.
Yeah, so we have done longer training sessions, and it seems like as we're doing the longer ones, they're able to basically stay and maintain kind of the performance gains. But once we stop stimulating the actual task, they do lose that so far. So that is like an active area of work, both in our lab and also a lot of the labs in general, is to get these longer changes. Maybe there's something going on with the culture that resets the kind of behavior and the policy of the network, but something to explore and find out. Yeah.
Thanks. Thank you. Next, we have from David Yekel. Hello, David. Truly amazing work. Your analysis relies on direct targeted stimulation of previously identified cells, which seems central to interpreting the results. Could you comment on how the results might differ if stimulation were instead applied to individual sites at predefined amplitudes without prior cell targeting? In particular, how important is targeted stimulation for achieving such interpretable and reproducible outcomes?
Yeah, I'll take this. So
Yeah.
the way that we can kind of see this, and this is a whole area to explore that I'm pretty excited to look into, is that when we were trying earlier, we really wanted to make sure when we were like specifically stimulating a cell, we get that one to evoke. But we've also tried experiments where we're stimulating them under the threshold of amplitude it would need to actually evoke those specific cells. So as long as we're hitting nearby cells, it seems like we are able to propagate and modulate the activity of the network. So I imagine it would work, and we can get to like kind of a more of a population scale and maybe get into the kind of like spatial area of targeting certain areas and reading out from other certain areas. So I would imagine and hope it would work really well, but definitely a lot of exploring to do there. It'll be tweaking the amplitudes and sweeping over all that stuff too. Absolutely.
Yep, yep. And because the organoids are dense anyway, so you would expect that there are always cells that you could stimulate. Yeah.
Yeah, assuming I guess yeah that you have actual tissue under them.
Yes.
So
Yeah.
yeah.
All right. Next, from Gillian. Hello again, Gillian. Does each organoid have to be trained uniquely, or were you able to create a generalizable automated training setup for identifying yours to stimulate, etc.?
Yeah,
Ash,
so
you have
we
many
actually
questions.
yeah, we did create basically an automated setup, and we have an automated thing going on right now. One of the last pictures in the paper I didn't share here basically shows that we could predict the capability of how like well the performance would be based off of those causal connectivity metrics I shared. Um, so the first order connectivity specifically being one of the more important predictors of how well the future performance was. So we've been kind of playing around with using that connectivity and other things in order to kind of guide the prediction, but nothing's like 100% learning or anything yet. But
Yeah.
trying to get there.
Yeah, thanks a lot. For our audience, we are going to go over time a bit, just because we want to cover as many questions as possible. And if in case not, we will be answering the questions by email, and send it to you later. But I'll go ahead. Okay, so next, from ASG. Interesting presentation. Thanks. Do you think the 3D structure of the organoids is important for this task? Could 2D cultures also do this? I guess any one of you can answer. Yeah.
I can go for a second, then Hunter can
Yeah.
go as well.
Yeah.
Um, Realistically, I think it's something to explore. I think 2D cultures probably have the capabilities, but the 3D seem to have a better ability of propagating activity through and like kind of richer dynamics. But also, Hunter, if you want to, or Silas, if you want to enter at anything.
Yeah, I was going to say this was point of contention for our paper as well. And we have some unpublished data that basically shows that the differences in the network structure is much less divergent when we have the 2D cultures compared to the 3D. So it does seem like the 3D structure allows a difference in information propagation. And how directly quantifiable that is when we translate that into learning or these other things is, definitely something we'll look into more.
Thanks a lot. Okay, next question from Jisoo. Hi, Jisoo. How long can you measure the signal continuously? Does the system have the incubator function? I guess like just how the setup looks like. I think you did not expand a lot on that. Yep. Who would like to answer?
Yes, I can go.
Yeah.
We right now we use the Max 1 inside the incubator. Generally, I mean, we use the Max 2 as well, but for
Mhm.
most of these experiments, we have done experiments on the order of a few days even, which is great, but also generates a ton of data. And then, I mean, I think one of our longest experiments, we had it scheduled basically to run on the hour every hour for I don't know. I think we've gone over a week before. So we've gotten some really rich signals, obviously, of like feed the organoids in between and all, and that is its own thing. But yeah, so really long amounts of time. Unless you guys want to add any more.
Hunter, Sebastian, any
I
other?
think
Okay.
you covered it. Yeah, that's great.
Yeah, it's all good.
Yes. Anyway, for our audience, if you would like to follow a lot more of the Brain Engineers' work, please go to their website. There's a lot more there. Yes. Okay. From Frederick. Hi, Frederick Seibd. So his questions are, he has two questions. I'm just gonna combine them. How are the organoids responding to pharmacological blockade of GABA, for example, precodoxin or gabazine? And as a follow-up, what do you do then? What do you then do with a trained organoid? I guess he's curious what happens afterwards. Yeah, I guess he's for you, Ash, and to the others if we also did some pharmacology.
I can answer the second part, but I think one
Yep.
of you guys should probably take the first one. But what we do with the trained ones, I mean, we generally just kind of keep training them and see how well and long they kind of go on and like kind of assess all their capabilities there. They don't have anything super special going on after the fact. Sometimes we
Thank
section them, but yeah.
Hunter and Sebastian for
So
the
so how we actually came to kind of the hypothesis and narrative of our paper was we had our dorsal forebrain, which is kind of the standard recipe for looking at how cortical assembly, whether that's molecular or electrophysiological. And when we added in gabazine, as others have, it increased the synchrony in the network. So that kind of led us to see a more natural and biological signal of perturbing that excitatory to inhibitory ratio. And then it is then as we're looking more of this goal-directed learning, we're looking at all of these different pathways and trying to dissect what is necessary and sufficient for the learning in that regard.
Yeah, I believe that like adding all the chemical perturbations are really important as controls for the experiments, and also can give you hints of possible mechanisms of the functional results that you are getting from your cultures.
Thank you, thank you. Okay, we have a few more. Let's go straight ahead. Next is from Paige. What made you select a 45-minute rest period between trials? I think that's for you, Ash.
Yeah, part of it was to basically like see I mean, we've explored different amounts. There's kind of a few factors here. One's like the amount of data that we can kind
Mhm.
of actually output and deal with there. The other one is like seeing if we've done some stimulation experience constantly on cultures, and when we repeatedly stimulate them a ton nonstop over time, we see changes in the activity that seem to respond a little bit less to the stimulations and things along those lines. So it is a I'm not going to say it's fully arbitrary as the 45 minutes, but it's a little bit arbitrary on both sides there. We just kind of had to select some parameters as well to go forward. Yeah.
Thanks. Again for you, Ash, from Jeff Gruen. Hello, Jeff. Is goal-oriented learning in cortical organoids plug and play for what you have done so far in the Maxwell API, or does it require additional programming and hardware? At least for what you have done so far, and then maybe from our side, I can answer.
Yeah, so for everything that goes on here, it realistically just is based off of the like all the Maxwell API and the hardware, but then we have our own package that is about to be fully released and pushed out open source that basically just wraps on top of those and makes it really easy to interface with everything. So in a perfect world, in hopefully a short bit of time, you'll be able to just basically pip install Braindance and have your Mac like on your Maxwell computer, and everything will just run and flow easily.
Thanks so much, Ash. And from us here at Muscle Bias Systems, so we really like our goal is to provide the tool for all of you, our audience and also for our speakers today with a device and the capability to actually interface algorithms and the biological part. And the API is very, let's say, available for you to really explore as much as you can with the hardware. And yeah, for our users, they're developing these new packages that you can also download and try out for yourself. So let's wait for the brain dance to be out and you can try it out yourself. And from our side, that's why we have also this neurocomputing focus this year. We also have tutorials and other simple to let's say starting scripts for you to use and learn about our API. So please contact us directly for any of these specific interests that you have. Okay, next is from Fitz. How long do your organoids grow for before they die, or can they be cultured indefinitely? Also, why do you use LDN in the ventral organoid protocol but not in the dorsal protocol? Hunter Sebastian?
We observed that the organoids start decaying in activity around day 70. It depends on the cell line as well of mouse that you are using. But that's something that we observe and was something that we also found out unexpected because you have seen that, for example, human organoids keep like being cultured even for 7 years. So that's something that also we're interested to look ahead because if you stimulate them, the ones that were trained by Ash, they tend to live longer. So
Mhm.
that's part of the interesting stuff that we also have observed. And about the LDN in the ventral organoids, it was more like kind of legacy that we got. LDN is just a BMP inhibitor, so of course it is required to have the validation of the case, but I don't think that if you add LDN to the ventral cultures or the dorsal cultures, you are not going to have the correct cell type. So it is just going to even improve more the purity of the tissue.
All right, thank you so much. I guess now I'll just go through three more questions, and we'll close the session if that's okay. And for the rest, we'll answer by email. Next, we have from Kioma Okimbata. Hello, Kioma. In your experimental case, which feature extracted is most informative for the network level analysis? How do you ascertain that activity patterns observed biological patterns and not recording bias?
Yeah. So for what we were looking at in our paper was trying to have a metric for that can replace connectivity, as we don't have the ground truth connectivity as you would in, say, an artificial neural network. So that was STTC is kind of a proxy for that, and then we use the statistical thresholding and as a way to look at high confidence connections. It's correlations in our paper, as you can't really say for sure whether they're connected or not. So it's kind of the best we can do at the moment. But one of the cool things that Ash showed in the paper was that using the causal connectivity instead of the spike time tiling was a much better metric for how well it's learning. So it can, using different ways of looking at the how the network is functioning by, say, combining electrical stimulation and probing the cultures in that way are can be better features and things that we're actively exploring. In our paper, we look at general burst structure and other metrics as well. So that was kind of the SDCZ was the main one that we use for building the networks in this case, but there's many that, depending on your use case, is more than, yeah.
Yeah, just
And then,
adding a little bit, STTC, well, there are different things that can also vary through the recording. STTC, it is still a metric that can be biased by firing rate. So that's why having this surrogate analysis with more than 1,000 shuffles and also hopefully 1,000 from 500 to 1,000 surrogate networks that are similar to your network, like the original network, but they are just shuffled. That helps you to create like what may be the significant pairs more than just like synchronicity by randomness.
and
All
then
right.
the second question with the recording bias, we used a lot of samples. So if that was one of the points in there that we recorded from over 30,000 neurons for I can't remember off the top of my head, but it was over 50 organoids for sure.
Wow.
So we had a lot of different samples to overcome any organoid to organoid variability or recording to recording variability in that sense.
Amazing. That's really a lot. Congratulations. Two more questions. So I think for you, Ash, you also spoiled a bit from your presentation beyond card pull. This is from Eric Arkfeld. Have you attempted any other control tasks?
Yeah, I mean that's the very interesting thing. We've attempted a few various ones, but we're in pretty preliminary area for all of those. But it's been a lot of testing to make sure that Braindance can kind of easily interface with all these other tasks, which so far everything's been going incredibly great. So everything we've tested, it's been able to interface and go really well. Should I guess also mention the Braindance paper is going to be coming out pretty soon as well.
Looking
And then
forward. And very last question, are you planning to implement optogenetic means of stimulation from Attila?
yeah, we actually have a we have optogenic capabilities. We have a previous paper that was in Nature Neuroscience and all about basically using human brain slices with epileptic activity. It was hippocampal slices, and we were able to basically shut down seizure-like activity with optogenics there. So that was with an early version of BrainNets as well. So we're absolutely exploring that whole pathway and tons of other things too. Yeah.
And yeah, thank you so much, Hunter, Sebastian, and Ash. Really, really, really exciting and very dynamic discussion that we had. And looking forward for the next work and research on papers coming out from the labs. And congratulations again, and hope to see you soon, maybe in the future events. Thank you, and thank you so much for our audience for staying until
Yeah.
now.